And now we'll move to Professor Chris Denton, again well known to most of you. Chris, one of our vice presidents, leads probably the largest scleroderma program at the Royal Free in the UK, and I think also very importantly currently leads the clinical group called the UK Scleroderma Study Group, which meets regularly to understand the priorities for treatment and research in scleroderma. So he's the ideal person to give us an update on recent clinical trials and treatments for scleroderma and Raynaud's. Chris.
So thank you, Jeremy. It's a real delight to be here. I know we are running a little bit behind. I'll try and make up a few minutes if we can, so that you get a coffee break. But I should say my journey in scleroderma isn't as long as some of the others in the room. I came into scleroderma in 1991, at the very beginning of 1991, when I was working at the Royal Free. And like Carol, I really appreciate this was a disease that was very challenging. And what I want to share with you today is really some of the progress that we've made. But it is a challenging title to talk to, clinical trials and effective therapies. And the very fact that we can have a talk about this, I think, is testament to the progress that we've made, and you'll hear more about that later today. So what I'm going to try to do is just review something that will be very familiar to many of you, the sort of challenges we have in scleroderma, but in particular how we're currently treating the core aspects of the disease, the fibrosis that occurs in the skin and the lung. Of course, scleroderma is much, much more than that. I haven't got time to go into some of the progress and treatments for specific aspects of the disease. I'll talk a little bit about pulmonary hypertension as an example, but renal crisis is a challenge that we can address, I think, much better than we could, and digital ulcers and other vascular complications, and some of this will crop up, I'm sure, in the other sessions today. Because what I'd like to just share with you is also some of the concepts that are moving forward in terms of perhaps getting to the next level of treatment for scleroderma and achieving in scleroderma what we've achieved in some other medical and even rheumatic diseases over the last few years, of really effective therapies that can really transform the disease. But before I go into that, I just want to highlight that this is something that's really an important learning point, I think, for managing any complex disease, that you don't just need magic treatments and effective therapies. You need very well organized care, and you need to be thinking about how to look after patients. And what we were able to show in this paper that I wrote with Carol is that if you just organize your care of scleroderma patients better and are proactive and look for the complications rather than just waiting for things to happen, you can improve outcomes. And this shows how people now are living with scleroderma rather than necessarily dying from it, particularly with the worst forms of the disease. And this is probably because we are vigilant in identifying particularly lung complications at an early stage when we can start to intervene, recognizing the treatments we have may be relatively limited, and I'll come back to that. So an important point of management is simply to systematically assess your scleroderma patients, and those of you that come to the unit at the Royal Free will be very familiar with the fact that every year we are really assessing whether you might have lung problems or heart problems or kidney problems, and really trying to be as systematic as possible so that we can identify aspects of the disease to treat. And this has become more formalized, and I'm really delighted, again, representing the sort of, or reflecting the progress and the priority that scleroderma and diseases like scleroderma at least are now receiving, I think, that NICE has recognized the importance of scleroderma, and in conjunction with the British Society for Rheumatology, the BSR, we have developed some NICE-approved guidelines now, and this has been developed by the UK Scleroderma Study Group and by the expert centre in a very multidisciplinary way, including patients and stakeholders in the process, so that we've ended up, and this has just recently been published and is available now to all doctors and in fact to patients and any other healthcare professionals online, guidelines that tell you how to really manage your patients with scleroderma and how to, when you should refer them, and the different approaches that are available. And so this is the flow diagram that summarizes the pathway for patients, but it just emphasizes the importance of systematically assessing patients and making sure they have access not only to the treatments that might be for lung problems, pulmonary hypertension, and so on, but also treatments for the morbidity of the disease, the non-lethal aspects of the condition. And although we've got lots and lots of challenges still, at least we can start to try and apply what we have in the best possible way. So I think that is a step forward. But what we'd really like to do is to sort of move things to the next level and start to really give treatments that fundamentally change the disease. And this is really the concept of targeted therapy, which is applied to many different diseases, but I think is particularly appropriate to a disease like scleroderma, because targeted therapy for scleroderma can have a lot of different meanings. It can be, just as I've alluded to, this concept of targeting the organ that's affected or most affected, whether that's lung or heart or muscle or the bowel. So we can target the organs in scleroderma. I think what we would love to do, and I think we would think might make more impact, would be if we could target the disease process. If you like, this might be moving a little bit more towards the goal of a cure for scleroderma, if we could actually target the way in which the abnormal cells in scleroderma are working and try and make them work more normally, and I'll come back to that in some later slides. And then finally, and certainly from a biological point of view, it's possible if we could understand some of these processes, we could find that there are actually really profound and things that we could do to actually affect cells in scleroderma that could prevent the complications or help to treat the disease better. And I will show you some examples of that. But I was asked to talk about recent clinical trials, and I think a more general statement first is that I think the organization and better organization of scleroderma care and doctors and patients has allowed us to develop clinical trials better. And we've also understood that scleroderma clinical trials have their own specific challenges, but most particularly that when you're trying to make progress with trials, you can't use the same trial design to ask every question that's relevant. And so this is a complex slide taken from a recent publication, but it just summarizes the fact that I think we do now understand much better. If we want to test something in scleroderma, we understand that there is a process that starts quite small with just a few patients being treated, but treated in a way that we can try and understand whether the drug or the intervention is making a difference, and then eventually moving into the much more large trials that are much more conventional. And the most important thing about any clinical trial when you're really trying to demonstrate effectiveness is not just the intervention you're giving, but the control arm of the trial or the comparison, so that you really know that whatever you're doing in your new treatment is better, or how it compares with standard approaches or with a placebo, a drug that doesn't have action. And I think it took us a little while to relearn these important principles of clinical trials, but I think we have relearned them now, and I think that's why we're starting to see progress, particularly now, and I think it's an exciting time, as I will show you here, because we've got a complex disease with different aspects of the disease, blood vessels being affected, inflammation in the immune system being affected, and of course the fibrosis of scleroderma. But we also now have lots of potential ways medically that we can start to intervene with these different processes. And I've just listed here the different pathways that we could target in patients that might have beneficial effects. But that's not just a theoretical list, because in fact, if we look here, we've now got lots of treatments that are in the clinic for other diseases or new drugs that are being developed that do target these pathways, and that would be the sort of things we might like to test to see whether this would make a difference to scleroderma patients. And again, this is far from theoretical, because these blue arrows highlight, in fact, the drugs in this list that are actually in current clinical trials in scleroderma or its complications like lung fibrosis or pulmonary hypertension. So we really have moved to an era where I think scleroderma has grown up to the point that we are now doing clinical trials, not just academic trials, but trials with drug companies and with the pharmaceutical industry, recognizing the importance of the disease. But if we step back into the present and the reality of managing scleroderma patients at the moment, just for a few slides, I'd just like to show that not only have we got some potential for new drugs, but we've also got a better understanding now of which drugs are working and that the drugs that we're using are helping our patients. And this is, again, an important point in looking after scleroderma, particularly thinking about the skin and the lung. And I'm going to summarize skin treatment just in one slide, because the concept of treating the skin in scleroderma at the moment is really focused particularly on those patients with the more severe form of skin disease, those with diffuse scleroderma, which, as you will be aware, can cause tremendous inflammation and thickening of the skin that's affected. But we think that this thickening and fibrosis probably is at least in part due to the immune activation and inflammation that occurs early in the disease. And so for this reason, as rheumatologists, we've been very familiar with using drugs that can dampen down this immunological overactivity. And so these drugs would all be very familiar to rheumatologists as drugs that are used to dampen down immune overactivity. And these really form the mainstay of treatments for skin disease in scleroderma. We are very confident that these drugs do help the skin, but they certainly don't achieve what we really want, which is a cure for the disease. You're going to hear from Professor Tyndall later some very promising results, I think, from the use of very intensive immune suppression with stem cell transplantation and high-dose immunosuppression. And I think it's that sort of work that has really shown us that this is the right theoretical approach to scleroderma, but we need to find the best way of delivering treatments that are effective to the right patients and obviously with the minimal toxicity. And that's why it's so important to be thinking about other new approaches to immune suppression. But if we just come back to cyclophosphamide, a standard chemotherapy, it comes from the field of cancer, particularly from leukemias, but it is a drug that effectively suppresses the immune system. It's an important part of stem cell transplantation, as you'll hear, but even used in lower doses, it does seem to be helpful for patients with scleroderma. The best evidence, in fact, has come from a large study from North America. The first scleroderma lung study compared taking tablets of cyclophosphamide for 1 year compared to placebo, so a tablet with no possible medical effect. And when cyclophosphamide was used in this way, it did show benefit. You can see here, this is an improvement in the lung function, so the lungs are expanding better and functioning better in the patients who've received the cyclophosphamide compared to those which really don't show an improvement on the placebo. However, of course, this is just marginally improving the lung function. It's not transforming the disease, but it is confirming that this drug has some effect on the disease process. But oral cyclophosphamide is quite challenging and toxic, and so in the UK we undertook a study of infused cyclophosphamide given as a drip treatment once a month, and we were able to show that in this study we really achieved the same benefit but with a less toxic approach. Again, so an improvement if you are on the cyclophosphamide and a worsening if you're on the placebo in this landmark clinical trial published just over 10 years ago now. So, but again, this is clearly a treatment that is helpful but not achieving what we need to achieve, and one with some toxicity. We were always very interested in the potential of a drug called Cellcept or mycophenolate, another immune suppressive drug used in transplantation that we felt could be helpful. And indeed, we showed when we looked at our scleroderma patients that patients who were on Cellcept did seem to have a lower rate of development of severe lung fibrosis compared to those that were on other immune suppressive treatments. And in fact, very recently, in the last month, the second scleroderma lung study from the United States has been published, and this drug has shown that mycophenolate mofetil 4, so Cellcept, is probably as effective as oral cyclophosphamide for patients with lung fibrosis. And in fact, using a slightly different trial design, they've been able to show that there's actually quite a clinically significant improvement. You see a nearly 10% of improvement in lung function over 2 years with the mycophenolate mofetil treatment, similar to cyclophosphamide, but with much less toxicity. So there's no doubt that MMF is a useful treatment, but it doesn't cure you, and it may not work as well in all patients. So there's always a need for more intensive treatments, and Professor Tyndall will talk about stem cell transplantation. I did want to highlight that this is a treatment that we've taken on board in the UK, and part of our NICE accredited guidelines has identified that there is a group of patients who definitely should be referred and considered for this treatment, and this is the case, and we've now got an active program in our centre at the Royal Free, and there are other centres around the UK that are following this up, but it's, as you'll learn, not a treatment probably for every patient, and certainly not without toxicity. So that's really the standard of where we are for treatment of skin and lung disease, focusing particularly on immune suppression, dampening down the immune overactivity in scleroderma, but hoping by doing that we can influence the blood vessel problems and the fibrosis that occurs. But if I now take a few slides to look further forward and think about that dream of getting a more targeted, effective therapy, perhaps with less toxicity, here's a cartoon that really summarizes the complexity of the biology of scleroderma, the involvement of the different cell types related to blood vessels, to the immune system, to fibroblasts, and to the connective tissue. And this is a busy slide necessarily because it's a complex disease, but what we can start to do if we provide this sort of roadmap of what might have gone wrong in the disease is that we can start to think, well, what could we do if we wanted to, in a more targeted way, block the disease or reverse the problems. And one thing we could do is to try and target the individual cells that are players in the disease, the cells of the immune system. So there are cells that are involved in making antibodies and cells that are involved in responding to tissue damage and causing inflammation. But it's these antibody-producing cells that are perhaps the ones that we could think most feasibly about targeting in scleroderma, because this is an approach that is already in use for other diseases, especially for rheumatoid arthritis, where we've got drugs that can block antibody-producing immune cells or B lymphocytes. So there is a drug called rituximab that does that, and there are some encouraging results from treatment of patients with scleroderma, particularly with scleroderma lung fibrosis, using this approach. And I just want to show you here just some results from one paper that we published in collaboration with Professor Wells and colleagues at the Brompton Hospital that showed that at least in a group of patients that had failed all of the standard treatments I've shown you with cyclophosphamide and mycophenolate, and still the lung fibrosis was getting worse or not improving, that in fact there did seem to be a change after these patients received their rituximab infusions that showed improvement in their lung function. Now, this is a very selected group of patients, and so this is something, again, that needs to be tested in a clinical trial, but it may well be that this is something that's particularly valuable, maybe for patients with lung fibrosis in scleroderma. So there is a trial called the Recital trial, ongoing now, led from the Brompton Hospital, but across the UK. We're all very supportive of it to test whether giving rituximab might be better than giving the intravenous cyclophosphamide, the control arm of the study. So it's going to be a controlled trial, and it will take time, but we'll hopefully get the answer. The other thing to think about is not just to target the cells that have gone wrong in scleroderma, the fibroblasts or the immune cells or the blood vessels, but to target the communicators from those cells, the proteins or the factors that those cells make that communicate with each other. These are called cytokines or growth factors, but they're really the language of cells, the way that cells talk to each other, and it's very likely that those growth factors or cytokines have gone wrong, if you like, or are produced in the wrong place at the wrong time. And so it's attractive to try and target those growth factors, and I'm going to show you two examples, but again, this is something that is definitely an effective strategy for treating complex disease, because again, from other areas of rheumatology and from cancer and other diseases, we know that targeting growth factors with antibodies, for example, can really transform the disease. So it's important, of course, to choose the right targets, and I'm going to show you two targets that have been chosen in scleroderma. So the first target is a protein called transforming growth factor beta, TGF-beta, and this was chosen because in the laboratory this is a factor that really drives fibrosis. It makes cells produce too much collagen, and it's a very strong driver of fibrosis in the laboratory. And we know that TGF-beta is increased in scleroderma, so it made sense to try and target that. The other growth factor I'm going to talk about, I'll come to afterwards, is an immune-derived growth factor called interleukin-6. But I'm going to focus first on TGF-beta. So TGF-beta is produced at increased levels in scleroderma, and we've known that from our research, and others around the world had shown this. And so it was very interesting to know whether we could identify that TGF-beta could be a factor that might usefully be blocked in scleroderma, because we know that particularly patients who have early stage scleroderma have evidence of high levels of TGF-beta activation, but at later stages when the patients have stabilized or improved, the TGF-beta levels are much lower. And you can see here all of the factors that are controlled by TGF-beta in a scleroderma skin biopsy. So we started this journey a while ago, and in fact 10 years ago we published the first paper of trying to block TGF-beta in scleroderma. This was a somewhat disappointing paper because the first thing we'd done was to develop an antibody that blocked TGF-beta, but only blocked it in quite a weak way because we were concerned that blocking it too strongly might be toxic. So we undertook a study, just as I showed you, with a control group of placebo-treated patients, and in different doses of the anti-TGF-beta antibody. You can think perhaps with the eye of faith there was a trend for the skin. This is measuring the skin severity in scleroderma to maybe improve more with the higher dose, but there was no really significant difference from those patients who received no antibody at all. So we couldn't say this was an effective therapy. However, we didn't lose our interest and we carried on working on this. And in fact, a group in North America led by Bob Lafiatis in Boston has recently reported a small study of a much more powerful anti-TGF-beta antibody that blocks all of the TGF-beta activity in the skin. And here you can see a much more convincing fall in the skin and improvement in the biopsies of the skin in this study. So it remains to be seen whether this is safe, but it certainly may be something that could be used to help scleroderma skin disease. But I want to just highlight another targeted type of therapy with the same concept, but targeting a different protein called interleukin-6. And the reason why I wanted to say this is that, in fact, this has moved much more rapidly into an area where it may be moving and soon be potentially something we could even be treating patients with bad scleroderma, because on the background of some research that Dr. Voon Ong and colleagues had undertaken at the Royal Free, we identified that there was a subgroup of patients with scleroderma that had high levels of this protein IL-6, and that was important because a treatment is available, tocilizumab, a drug that blocks IL-6 that is already in use in medical diseases. It treats rheumatoid arthritis and other medical conditions. And so we knew that if the IL-6 was expressed in the skin in relevant places, that blocking IL-6 could be helpful. And we knew that patients who had high IL-6 levels did badly. And so this was something that might be relevant for a particular group of patients with high levels of IL-6. So we took this idea, in fact, to the company that makes the antibody treatment, and to be a bit cliched, they liked the idea so much that they took the whole thing over, but that was in a good way, because they went on and did a study called Fascinate, which just reported earlier this year, that where patients, again, were treated either with this tocilizumab treatment or with a placebo over 48 weeks, so nearly a year of treatment. And what you can see is, just as we might have predicted, those patients who are on the drug blocking their IL-6 showed an improvement in their skin disease, and those patients on the placebo really showed a fairly stable pattern. And so this, again, does point towards benefit and was sufficiently convincing. There's now a larger clinical trial that, if positive, I think would lead to this drug potentially being available. But I should again caution that this is very unlikely to be a drug that is the treatment of choice for every patient. But if you're in that group of patients with high levels of IL-6, it may be very helpful. There was also the signal, which was somewhat surprising, but again gave us more confidence about this approach, that we might even, rather unexpectedly, have been preventing the development of lung fibrosis in scleroderma, because, again, those patients who were on the placebo treatment showed a higher risk of developing impaired lung function. So in fact what you saw was that only 1 in 10 patients showed a worsening of lung function over the duration of the study if you were on the active treatment, but 1 in 5 if you were on the placebo. And this was an interesting finding, because it may suggest that you could even be preventing the development of a complication. So that's exciting, and that's really where we are, I think, in terms of trials and excitement in terms of treatment for the skin and potentially for the lung. But I just wanted to finish just with a very few slides to highlight progress that's occurred in another area, in particular the changes over the last few months or in the last year or so in our approach, and that is for pulmonary hypertension. So pulmonary arterial hypertension affects about 1 in 10 patients with scleroderma overall, but is a complication that used to be unfortunately always fatal. Now it is something that can be treated, and there are drugs to treat it, and in fact there are multiple drugs to treat pulmonary hypertension now, and you may recognize some of these, and they target different biological processes: endothelin, nitric oxide, prostacyclin. All of these factors seem to be important and disturbed in scleroderma and pulmonary hypertension, and they can all be normalized with treatments. When we first had these treatments, we tested them in our scleroderma pulmonary hypertension, and we were encouraged that we saw some signal that they were helpful, but it was not clear that they were really transformative. And here this was the very first trial of an oral therapy for pulmonary hypertension, showing just the scleroderma patients in the clinical trial. And what you can see, if you remember back to that last slide with tocilizumab, those patients who were on the placebo got worse, and those patients who were on the active treatment seemed to be stabilized. But again, this was less than you really needed to achieve. Well, if we fast forward now to the modern era, much more understanding about pulmonary hypertension and about clinical trials. And so in fact now, rather than just seeing whether you can improve patients' exercise capacity over a short time with a treatment, we now look to see whether you can really improve survival and complications and progression of the disease in a meaningful way. So time to clinical progression in pulmonary hypertension is now the way that trials are done over a long period of time. So here you can see data from a trial published a couple of years ago now of macitentan, an endothelin blocker, but showing that you can really over 3 years do much better if you're on the active treatment compared to the placebo. But more importantly, about a third of the patients in this study had scleroderma, and likewise for a very recently published study, the Ambition study, showing the same pattern of benefit, that if you give 2 drugs rather than just 1 at the beginning of your treatment, you seem to do better than if you just have 1 drug alone. So it comes back to the point that the more intensively you treat the disease, the better the patients do. And the point of this is that now, I showed you that early study where scleroderma patients seem to be responding to treatments for pulmonary hypertension, but not as well as other patients with pulmonary hypertension. We've moved on from that. So in these recent studies, I showed you the Seraphim study. That was the study of macitentan. There's a study of another drug called selexipag, and then the Ambition study where we compared whether patients treated with 2 drugs rather than 1 at the beginning did better. You can start to see here that in fact the patients with scleroderma are doing just as well from these treatments as the patients with the other forms of pulmonary hypertension. So it really does tell us that it's a complex disease, but when you've got multiple ways of treating it, if you combine them together and give them more intensively, you can probably achieve more. So I think I've finished there, but I just wanted to hopefully have communicated to you really that systemic sclerosis clearly remains a huge unmet need. It remains a rare disease, what's called an orphan indication, something that doesn't probably get the investment and attention it should do. But we are making progress, and part of that progress is not just the new drugs we have, but also the better ways we have of testing them. And I think we are in a situation where we've got now a growing number of treatment options that we need to make sure we're applying in the right way. And part of that is having evidence-based guidelines for treating patients so that hopefully patients wherever you are in the UK can have the same high level of treatment. So I'll just acknowledge the large number of people who many of you will know who work with us at the Royal Free and associated institutions. And if there is time, I'm happy to take some questions. Otherwise, I am around all day if anyone wants to come and find me. Thank you.